Ok, buenas tardes a todos. Estoy encantada de presentar a mi amigo Olivier Tenaillon. Nos conocemos desde hace 20 años y ha sido increíble conocer a Olivier porque piensa de una manera totalmente diferente a la mía y eso es fantástico. Lo conocí al final de mi primer sabático cuando estábamos en Irvine con el doctor Brandon Gaut y su mujer estaba también con Brandon haciendo un postdoc, mientras que Olivier trabajaba en San Diego con el doctor Lin Chao, aprendiendo modelos de mutación en virus. Y luego nos volvimos a encontrar otra vez en Irvine en el siguiente sabático, o sea, 10 años después, él ya haciendo un sabático y Maud también. Y en ese sabático él estudió los patrones de mutación en un experimento a largo plazo en E. coli, el experimento de Lenski, pero en este caso era con genómica, viendo los patrones de mutación en los genomas de E. coli que habían evolucionado en diferentes regímenes de calor. Entonces, ver cómo las hipermutantes se volvían hipermutantes y Olivier, desde muy joven, estudió en el Polytechnique, lo cual son escuelas de muy alto rango en Francia, estudió física y matemáticas. Entonces, realmente entró en el doctorado a entender a las bacterias E. coli, viendo tasas de mutación y modelándolos, y desde entonces se ha dedicado precisamente a eso, ver cómo E. coli se adapta en vitro y en vivo y cómo puede sobrevivir, sobre todo ante toda la crisis de antibióticos. Y entonces es un privilegio tener aquí a Olivier, un gran amigo, pero además un gran científico que nos habla de E. coli y sus genómicas. Bienvenido, Olivier.
Merci. Okay, so I will switch to English.
Yes.
Thanks for the introduction. So I'm going to talk about E. coli genomics. And so I work in the northern part of Paris, as we were discussing before. And here we have it's a place, it's a very popular district where we work in the hospital, and we're the heart of the COVID stories in France in that hospital. But through the years, what I've been working on in that hospital is E. coli, on which I started my career, as Valeria said. And this bacteria is interesting because it's a mother organism, and most of you may have cloned something into it. But it's also a common cell of vertebrates that transit from the gut of different vertebrates to the soil. And as such, we can sample it super easily. And Valeria has been one of the pioneers of sampling E. coli all around. But it's also a pathogen and involved in many forms of disease, from tourista, diarrhea, to blood infections and urinary tract infection. And through those, especially nowadays, through those extraintestinal disease, it's suggested that it kills close to 1 million persons a year. It's a genetically structured species with some different groups, and those different groups have different meaning in terms of their virulence and resistance. And actually, I've been working over the last 20 years with Eric Denamure, who's been doing epidemiology of E. coli, and especially in France. And he's been studying clones of E. coli throughout time in France, and we've studied samples from the 80s, the 2000 and 2010, and we are now doing the 2020s. And what you can see here is that there's different phylogroups that have been defining the species, and you can see that their prevalence has been changing in France, and particularly this B2 group has been increasing. And there's also, alongside, an increase in antibiotic resistance that has been observed, and you can see that over the years there's more and more strains that have become resistant to any kind of antibiotics. But also an increase in virulence, because this B2 group especially is loaded with genes involved in virulence, and actually those strains are the ones that are usually involved in extraintestinal disease. And as a result of their increase in frequency, we see in Europe, at least we see an increase all over Europe over the last decades. And we can see that there's more and more issues of infection due to E. coli over the last decades. And this is not the case all around, because those B2 strains tend to increase. They were already highly frequent in Japan and in the US since the 80s. and also in Australia, but in Europe it has been increasing, but elsewhere in the world, you can see that the frequency is much, much lower. You barely find them. We've been sampling in French Guiana among the Wayampi Indians, and here you barely found any of those B2s. So what's interesting is that we see that, in terms of epidemiology, we see that there can be some change that can be seen over a lifetime, over a few decades, we can see those changes occurring. And so one of the questions that we've been interested in is what are the drivers of these changes? So some hypothesis is that it may be antibiotic resistance, and the different strains may have different resistance, and we may promote the massive usage of antibiotics may have changed things. But also what we've seen is diet and hygiene has changed. In France, we may finally be clean and cleaner than we used to be in the past compared to Americans maybe, but also there's been a lot of change in diet and hygiene habits over the last decades in Europe. So resistance comes as an obvious candidate, but yet if you follow the epidemiology of E. coli through in infection, here this is work done in the UK, and they followed E. coli from urinary tract infection, and you can see that some clones emerged, and this one in green has been really a success story of a clone that rapidly increased, and this one is resistant to antibiotics. So it's part of the story that some clone may emerge because of resistance. But yet, all the other clones at the bottom are clones that are fully sensitive to antibiotics, and you see that their frequency has not changed that much. So antibiotic may be part of the solution, but not only. There's other factors that explain, that contribute to the difference, prevalence of the different clones in E. coli. So the idea is that we can sample clones, but the idea is, can we learn something from genomes? Because now it's with the the genomic revolution, it's way easier to have access to information at that level than to sample clones and strains and phenotype them. So there's been a switch, because in the past it was way easier to study the strains. Now it's way easier to study the genome, and now we have databases with more than 60,000 genomes or 100,000. I'm working with one with my hands at night on 60,000 genomes. So this database is already outdated because I sampled the genome like a year and a half ago, and now this same database is like, I think, 150,000 genomes. So there's a massive amount of information there, but we need to understand what's going on. And so for that, we need to know exactly what's the signature of adaptation in a genome, what's going on. And then we can try to see how genome evolve in natural setting, and eventually ask some question in the lab concerning diet, as we'll see. So the first thing I've been working on since my sabbatical and a bit earlier, as Valeria mentioned, is what can we do? Can we learn something from experimental evolution? You presumably all know that you can evolve bacteria in the lab quite easily, and nowadays we can sequence, the genome for about $20. And so it's easy to do that, and there's two objectives. First is descriptive, to see what's going on, but the second one is, from this repeating process, we can go further than just seeing, but trying to see whether there's some patterns, and whether this is the best case scenario for selection, because we know there's selection going on in those experiments. We can quantify it, and then it's the best case scenario to see what's going on in the genome. And so if we have to define some signatures of selection at the genome level. So for that, I'm going to talk about two studies that I've been involved with. They are, of course, the best in the field, not biased here, but no, they're interesting studies because of their scale. In the first one that I did during my sabbatical with Al Bennett, Tenaillon, and Brandon Goat was to study the evolution of 100 lineages that were adapted for 2,000 generations. At the time, it was the first time we had such a high level of replication with genomes that had evolved long enough to see quite complex patterns emerge. On the second hand, I've been involved in the study of Richard Lansky's lineages that were with Dominique Schneider and Jeff Barrick, and this time there's only 12 lineages, but those ones have been evolving for 50,000 generations. And so we sampled, in each case, we sampled either one clone at the end of the evolution for the thermal adaptation. So in the first evolution, the selective ratio was thermal adaptation, And in the other one, it's a long-term adaptation to limited resources. And here we sampled 2 clones at 11 time points throughout the evolution. So what I'm going to go fairly brief because these are quite published studies. But the idea is to come out and to see whether there's some patterns that we can appreciate at the genome level that tell us something about adaptation. One of the first observations on which I started my PhD is that when you sequence the genome now, you can see that among the 12 lineages, if you follow the number of mutations through time, you can see that here, 6 lineages out of the 12 have suddenly an increase in the number, in the accumulation of mutations. The 6 others, there's a zoom here, they have barely 100 mutations after 50,000 generations, while the other one have, hundreds to thousands of mutations. And what happened here is that we select for a high rate of mutation. These lineages are called hypermutator, and I've been showing during my PhD, and others have shown that when there's many beneficial mutation, you can select for an increased mutation rate, and that's a way for to adapt slightly faster. So that's one signature of selection, because when there's many beneficial mutation, when there's adaptation going on, we often see selection of mutators. The second thing is that we can do, so first was rough numbers, and we see huge difference in numbers. The second one is to look at mutations in the genome. And one thing we can do, after we counted mutation in the previous part, now we can categorize the mutation. And one thing that is classically done in genomics is to categorize mutation as non-synonymous and synonymous. The idea is that synonymous mutation, because they do not change the sequence of the encoded protein, have almost no selective effect. Of course, it's wrong because everything is wrong in biology if you look carefully, but at first order, let's say it does hold. On average, synonymous mutation don't have much of an effect. And if they have an effect, it's only a tiny fraction of them. Yet non-synonymous mutation are supposed to be subject to selection. And so the idea is that if we differentiate the two, if we make a ratio between the two, that could tell us whether mutations are selected or not. So the synonymous mutations should accumulate according to mutation rate because they're not subject to selection, while a non-synonymous mutation should accumulate through mutation, but also be filtered or selected by natural selection. So this was obvious. It was not super clear what should come out of those ratio, because on the one hand, if population is adapting, you should expect some new mutation to occur. But at the same time, if selection is efficient, you should expect a purge of non-synonymous mutation. But what came out in most experimental studies is that, as you can see here, when we do this ratio and work through it through time, we can see that early on, there's 16 to 30 times more non-synonymous mutation than expected. And this is a very strong signature of selection. Even after 50,000 generations, we find a large excess of non-synonymous mutation. Why? Because when the mutation is selected, when a mutation is neutral, it takes a very long time to accumulate. But when it's selected, it occurs and invades the population fast. And so as a result, you have this excess of non-synonymous mutation. In the thermal adaptation that we run with the 100 lineage, we find also a K of a case of about 5. So this is a common signature in all experimental evolution. We find that this ratio can be used as a signature of selection. It's interesting because you just mean one population. You don't need replicates. You don't need to measure fitness. If you just measure this ratio and find it's high, it tells you some adaptation has been going on. Then, after we looked at numbers and categorizing mutations just at a very low level of description, synonymous and non-synonymous, we can look at where mutation land. And we can find whether they land in the same gene or in different genes. And what we observe is that mutations tend to fall in the same genes, meaning that here you have, for instance, this putative transporter, YBAL, and every bar or symbol here represents that one lineage has a mutation here. It can be an insertion, a deletion that is in frame or not, or a point mutation. And you can see that even in this mutation here is found in 4 different lineages. So when you have enough replication, you can find that clearly this gene is selected to be inactivated in this experimental evolution because you have lots of frame shifts and eventually deletions. And this is something we can quantify. We can quantify the level of convergence. We see it, of course, in gene inactivation, but we also see it in essential genes like the RNA polymerase, where here you have one codon in which you have more than 10 mutations within one codon. So we can quantify that, and we can, through that, identify also some functional targets. For instance, here, there's a subset of genes who are involved in cell shape. Genes MRDA, MRDB, ROTZ, MREB, MRC, MRED, all of these genes, if you inactivate them to some extent, the E. coli that is, you know, as a rod shape is turning to a circular, a round shaped bacteria. And as a matter of fact, in the thermal adaptation, we can find that, you know, all of these genes are hit by mutations. Now we can quantify convergence. You can quantify it, for instance, by looking between two lineages, how many mutations are shared, you know, what's the fraction of mutations that are shared. And you can do that looking at specifically the mutation. Do they share the same exact mutation? Do they share mutation in the same gene, in the same operon, and the same functional unit? But you can see that the gene is convergence at the gene level. You gain clearly some insight. At the operon a little bit. At the functional level, you could, but it's also require much more work because you have to find out those personal units. And unfortunately, it has to be done through literature reading. So this is really time consuming and of course, subject to more discussion. One other thing you can do is to look also how those mutations are spread. So you find that there's convergence, but you can find out how are they spread between those different genes. So for instance, here it's a complex way to illustrate that, but here you have major targets of adaptation in the thermal adaptation lineage. You have ICLR, which is a regulator, CLS, which is a cardiolipid synthetase, Rho, a termination factor, the RNA polymerase, the sigma factor 70 RPOD, regulator for metabolism. All the genes I mentioned here involved in the rod shape, genes involved in the capsule, in the response to stress, the transporter I just showed you, and genes involved in metabolism. So they are clustered according to functions. And here, what you can see here, when there's a dot here, it means that there's a lineage that specific allele of CLS, one of the specific mutations that we found, and one in ICLR. Normally, there should be a line on the diagonal, but I took it out to show you that most of the boxes, except in the RNA polymerase, are empty. Which means that when you find one mutation, when one lineage has selected one mutation in one of these genes, there are not two. So which means that those mutations are not spread randomly. If you were to take all the mutations found in the experiment, you cannot just take them and redistribute them across in the genomes randomly. There are some subset of mutations that are found and exclude one another. If you have inactivated YBAL once, you don't need another mutation to inactivate it. It does also hold for essential genes like the genes involved in the cell shape. So this is a form of epistasis, meaning that one mutation does affect the selection at the other site, okay? And so what we found is that, you know, one mutation is enough, and those mutation within those clusters of functions or stuff are mutually exclusive. There's also some other form of interaction between genes. For instance, here we can compute some statistics also, and briefly just to show here that you only find genes in those clusters if you have a mutation in the RNA polymerase, which means that there's different paths for adaptation. And here in that system, you have one path in which either you pick a mutation in Rho and then you will not select mutation in those genes, or you select a mutation in the RNA polymerase and you further can have a selection and evolution through those genes. One other example of that is a work related to a work from Jeff Barrick, where here he's doing population sequencing of one of the lineage. And here you can see when you do population sequencing, you can re-infer the history of mutations. For instance, say that this mutation YBL occurred, it increased in frequency, and finally within genomes having YBL mutation, another set of mutants occurred and finally invaded. And you can see here a complex story where some clones eventually start to increase in frequency, but alternative populations of clones made it through and outcompeted them. What was interesting in here is that we observed that FABAR is here a mutation that occurs early on in the evolution, but we never see it any time later. So to study that more carefully, what we did with Alejandro Cauce, a postdoc in the lab who's now running his own lab in Madrid, what we did is we did a transposon-seq experiment. Transposon-seq is an experiment in which you inactivate all the genes in the genome, and through some very painful experiment, you can study how gene inactivation are selected or counter-selected. Sorry, I'm just going to close one window. Because my son's playing basketball outside. And so basically, you have the genes here, and you can see that a bunch of gene inactivation, when we do that experiment in the ancestor, are selected upon, including FABR. So inactivation of that gene gives a substantial benefit. Here, genes are ranked according to their position on the genome, and then the x-axis, you have their selective effect. Okay, so most of the genes are deleterious, but quite a few are beneficial. What's interesting is that after 2,000 generations, which means that in that genome, there's 5 mutations that have been selected for, and none of them are in FABR, you can see that now there's almost no more strong beneficial effect that can be acquired through gene inactivation. And when we looked at that, we can look at the benefit of mutations early on. We can look at, for instance, the 20 best knockouts early on, and you can see that after 2,000 generations, most of them are not beneficial anymore, including FABR. So FABR is one pathway that we don't know what would have happened if it had been selected for, but we know that the path that was taken by populations excluded that strategy from adaptation. Okay. So to sum up, it's a lot of information, but what we see as genomic signature of adaptation is that we can see that when there's adaptation going on, often we see an increase in mutation rate. We have systematically a high K over Ks. We find convergence at the gene level, which means that usually there are many mutations, even in essential system, we find many different genes, many different mutations, but few gene targets. All sorts, I haven't mentioned it, but also all sorts of mutation can be beneficial: point mutation, deletions, insertion, duplications, large, small, everything. We see many gene inactivation. Also, something I've not mentioned is that usually we see early on some quite global regulators. Some genes evolve that have super pleiotropic effect, like I show you briefly, the RNA polymerase, tend to be a major target of adaptation, or genes involved in supercoiling and all this stuff that controls many genes. And also we are able to see interactions. Yet when we do that, we have to keep in mind that presumably the landscape experiment is the most boring environment ever. Is it, imagine that for 20 years, or now 30 years, sorry, those bacteria have been having the same food at precisely the same time every day at the same temperature. Do you think it happens in life? No. And so it's not only boring, but it's also boring and weird, because if you look at, for instance, here, when we align 133 genomes and we align the coding sequence, the mutation nodes, and we plot the non-synonymous mutations observed in the RNA polymerase, we find there are 5 non-synonymous mutations in the RNA polymerase. It's the most conserved genes in the whole genome at the species level. Yet, after 2,000 generations of adaptation at high temperature, we have all these different mutations. So this environment is far from natural. So it can question the relevance of these observations. And so now we know that when there's selection, there is this signature, but we can question, is it true? So one way to see if it's more natural, what happens to wildest sample humans. However, this is more complex than expected. Why? Because, these are bad slides, but it's showing I'm involved in a protocol in which 22 healthy volunteers have been treated with antibiotics, and we've been sequencing some mix of E. coli strains, and colors reflect diversity, different genotypes. And basically what you can see here is the treatment. Is you can see that, initially there's a red clone, a blue and a green, and others, and eventually, after the treatment, the red would go away, the green increase slightly, the blue, and so on. So we've done that, but we're surprised to see that there are very different patterns. And what's surprising is that sometimes it's very stable, one clone that stays over 180 days. Sometimes it does change a little, and sometimes it does change within 2 weeks. And at least 30% of the ones we had, of the 22 patients, changed within 2 weeks, which means that to study what's going on and study mutation within one individual can be tricky because a lot of them will not have the same clone afterwards. So this is something telling us it's really relevant ecologically, but we have some question about the relevance of that. So now we need big numbers, and it's really hard to get those big numbers because those protocols, if you use a lot of healthy volunteers, it's super expensive. But François Blancard in the lab just got a grant to follow up to 200 people for several times. So we're going to have in the next future some data on that at a much larger scale. So as I say, it's not easy to have longitudinal data. Yet we had some in the lab. Actually, a lab member managed to follow these strains, and we find out that over 1 year, he had one dominant strain. It was the only E. coli found in his gut, and it stayed for a whole year. So we could sample at 3 time points, isolate 8 clones at 3 time points, and sequence the genomes. And that was work that was done by Mohamed Galliani and Adrien Launay. And here I plot in eBurst kind of format those results, meaning that here at the first time point you have some genome, and here when numbers are in the same genome, the side represent the number of genomes that have the same exact sequence. Here it's a genome that is 2 mutations away from that one, and the colors show the dates. So here you see that from time 0 to 7 months later, you know, presumably the population have moved from maybe those genotypes to that one, maybe one mutation emerged. And then over the next 7 months, the population fixed another mutation. So when we see what occurred over 1 year of evolution, which is equivalent to the 1 year we saw for the thermal adaptation, we saw that, you know, barely 1.5 mutation made through the population, and one is synonymous here, so not much is happening. So that was a bit disappointing, and we are not the only one, because another of the scientists studying E. coli is looking at his own E. coli, and here he followed not only his own, but his dog, his wife, and his daughters and son. And what he could see is that his sequence here, it was not a dominant clone, but a clone that he could find throughout the family for 3 years. And when the sequence, and you look at the sequence, you can see that over a few years, you know, for instance, samples that were sampled in 2005 and samples that were sampled 3 years later just differed by 2 mutations. If you make that pass, there were only 2 mutations. So what seems to be quite surprising is that there's not much evolution going on in humans. Okay, so no traits. So we see very limited evolution, few mutations, no, of course, no signature of selection. Yet we do not have much replicates, and we have no controls of what has been going on. So how can we go further? So one way to go further is to look this time at, you know, make a mix between full nature and experimental evolution in test tube, is to go inside the mice. Ideally, we would love to go to axenic mice where you control the full microbiota. Unfortunately, I mean, first, these are weird animals because you grow them partially for some time without any flora, so all the immune system is weird and so on. Plus, but the trouble is that here we want to do long-term evolution. So whenever I present this project to people, they say, "Oh, great, would love to collaborate with you with our axenic mice." Then I tell them that I want, you know, something like 10 cages to have replicates or 15 cages to have replicates over a year, and then nobody's answering my emails anymore. So here the idea is that to do that in a much cheaper way is to use streptomycin treated mice. Streptomycin is an antibiotic that is affecting enterobacteria, but not all of them, so that when you treat the mice with that, you clear the niche of E. coli and slightly more, but you still have a complex microbiota in the mice, and you can clean it, introduce the strain you want, and then keep treating with the antibiotic. So we have control of the strain we put in, and we have partial control of the environment because we can also control the food. The benefit is that here we have replication that is possible. So one of the questions we wanted to address is genomic evolution similar to the one we observed in the laboratory? And later on, what's the impact of the diet, but also what's the impact of the strain we use? And so first, we use one strain that we use for decades in the lab. It's a B2 strain. And what we observe is that over a year, we observe 7 mutations per genome, which was very consistent with what we had with experimental evolution. We observed that KAKS was higher than 1, as expected. We observed some convergence, some interactions, and some inactivations. Just to illustrate a little bit that, sorry. what we observed is that, for instance, convergence was very similar, about 20%, very similar. So two lineages that evolve in independent cages had 20% of their genes affected that were the same. We found mutation in some genes. I can discuss that later. We find some epistatic interactions, like some dependencies, like for instance, lineage had the operands for galactonate inactivated, activated in some way because the repressor was inactivated. And then in some lineage where the repressor was inactivated, we find mutation in DGOD. And in the one that had mutation in DGOD, they had also mutation in DGOT. So we see that, you know, there's a path here. If you have that mutation, you later on have that one and that one. Conversely, we have also here two mutations that are involved in streptomycin resistance, and you either have one or the other, never the two simultaneously. So this is consistent with what we had with some of the patterns we could see in experimental evolution. Yet what was different with experimental evolution was that the KAKS was way smaller than what we had. I told you about 32-fold to 5-fold, and here we're at barely 2. We could not find any mutators. There was no global regulators. All the functions were linked either to metabolism or, unfortunately, to the antibiotics we used. Unfortunately, because the strain we used was incredibly resistant to the antibiotic, up to a level where it almost resists to a puree of antibiotic. You know, it resists to 170 grams per liter of antibiotic, which start to be expensive to produce. But still, we could see some of the mutations were involved in that. No global regulator. And one thing that was quite surprising is that one of the mutations we found consistently was inactivation of the regulation of an operon. And it's surprising because this is a working repression. So which means that you can induce that in the presence of galactonate. The operon is functional, so we could test that. We could validate the system. And yet there's selection for inactivation. And actually in another experiment using young baby mice, we also find inactivation of a way famous LAC-I. And here also it's surprising because we think that, you know, having an inducible system is optimal, but here in both systems we found that here selection is favoring the loss of a smart system towards a constitutive induction of the expression. And we could find, despite the super high level of resistance to the antibiotic, we could find that the genes RLD and GB, which are weird genes involved in some complex systems, are not increasing really the concentration at which the strain resists, but they allowing the strains to go way better. Here's the division time in the presence of streptomycin, and you can see the single and double mutants are doing way better than. So we were excited because we found that it was a bit different, but then we wanted to test. We say, okay, we have a setting, and one of the questions we wanted to test is that, you know, the change in diet in France and in Europe and around the world has been to go from kind of healthy food towards a Western diet food, which is high fat, high sugar. And so we wanted to go and to try whether if we change the diet, whether the bacteria would adapt in a different way. So we used that strain, and to make things short, you know, the numbers were quite similar. When we look overall at the numbers, we find that no mutators, we find the same amount of mutations over a year, the same KKS, and a slightly lower convergence. The genes targeted were kind of similar. We find GB RLD. We find one of the gene of a non-function to a lesser extent, and except there was nothing this time, though, in two targets, KPTA and DGO. The operon I mentioned you was a big target for adaptation. So there's a bit of specificity to the diet. Unfortunately, galactonate is a media where we don't know exactly where it's going on, and it's super expensive to work with it because it's something like $500 a milligram. And KPTA is a gene whose description is that it's working on introns, and so it's not clear what it's doing in bacteria. But but there's a little bit of, you know, environmental effect. You know, the diet is, the E. coli is sensing that it's a different diet because it's adapting through different genes. So that was okay, but so we decided. We used so far one strain. Can we go further and use a different E. coli to see what's going on? And this time we choose a strain that from the group A, which is a group that used to be the dominant group in France, in commensal in humans. And here we are quite surprised because everything was different. So here we have much more mutations. We have mutators, and those numbers, do not include mutators. So in addition, we did 8 replicates, and we have 2 mutators in each that emerged. We have a KKS. I told you here it was 2, while in experimental evolution usually it was 5 or higher, and here we have 15 and 8, and we have some convergence at some level. Okay, so looking at mutation rate was surprising because even the strain HS has a lower synonymous rate, significantly lower, but way more non-synonymous mutation, way more indels, way more large deletions, way more IS transpositions, so transposition of insertion sequences. So every system of mutation has been recruited to increase despite presumably a lower rate of mutation. There's some convergence, we can work that out, and surprisingly we can see that here, while for West 536, if we do a PCA on the different types of mutation, we can see while for 536 the different diets were not that different. Here we find that HS, the 2 diets are different, and the mutations are different from the one selected in 536. So for instance, we have large difference. We have only mutation in GID-B and never in RLUD in HS. And we have difference in metabolism. So here I plot that. You can see here at the bottom, this is what we observed in E. coli in the B2 strain. Here, what we observe in this new strain from the group A. Here on the right-hand side is the CHO diet, the healthy diet, and then the left-hand side is the Western diet. And so what we observe is that here we have galactonate that was present here, but it's not at all present in this other strain. While maltose is present in both, but to a much larger extent. And there's a bias that we observe more in CHO diets. And here we found much more expectedly a lot of mutations involved in gluconate uptake. Gluconate has been shown in the past to be the main sugar for E. coli in the gut. And here we find a lot of mutation involved in that pathway. Yet, surprisingly, those mutations are mutations that tend to inactivate the transport of gluconate. So it remains unclear why. And that's an overall limitation of those experimental evolution, is that, finding out the mutation is possible, is cheap now. I mean, you evolve, which is painful, then you sequence, which is okay, and you analyze the genome, that's okay. But to go to the functional analysis, you can validate the effect of those mutations by doing some growth curve, and we can validate that those mutations are really a phenotype expected. Strains cannot grow on gluconate and here on ribose. But why are those selected? Why are those mutations selected remains a real challenge, because for every mutation, there's a specific story to be found. And I remind you that potentially natural selection can favor in the system mutation that increase by 1 or 2% the efficiency of a system. So understanding why is it good to be 2% more efficient in gluconate uptake, or here it's loss of function, so it's here, but why is it providing a benefit is can be really hard. So again, what we find out is that, again, we find consistently some loss of regulation. Here we find that mutation tend to favor a constitutive expression of some sugar. And it's been known actually that it's slightly better that even when you have fully induced, you still have a little bit of repression. And so it would be, and so it's driving the question of how could you maintain in the long term those functional regulation if in the short term it's beneficial to lose them. And so we're trying to look now among a natural isolate sequence of natural isolates whether we have traces of those inactivations. So if we look in strain HS, in yellow we have I think a Western diet and a CHO diet. This is different types of mutation. You can see, I mean, it's just a visual pattern that you can see that according to the 2 diets, you have different types of mutation that are recruited. It's different targets here and here. So we find some diet specificities in HS. And for instance, we find also in metabolism, but also in different genes. For instance, here, one of the 2 component system is mutated, where here you have, it's nice at the molecular level because you find that there's mutation in gene CPX6 that is supposed to activate CPX-A, and you find that the mutations are in CPX6 and in CPX8 in the sensor domain. So, and then you have also genes depending on that system that are mutated. So here we have trying to reconstruct the story of what's going on. Okay, so what I'm about to conclude here before I take questions. And so what are we seeing here? Of course, it's not a complete story because it's a never-ending story, but there's what can we see about the patterns of genomic evolution in vivo? So we find that surprisingly, at first we thought we had some similarities and difference, but here we find that there's a lot of similarities depending on the strain, and for some strain, basically adaptation in the mice gut is very similar to what we see in a test tube, which is kind of surprising because I've thought that those strains are really well adapted. So the main difference is that remain is that we find mostly metabolic targets and not some global regulators like RNA polymerase. So one of the issue and stuff I've been working on at another level is that the one potential, and we're going to try to test that, is that eventually one strain is way more adapted to the gut than the other. Okay, and presumably like this A strain may be less adapted to the gut, and therefore when you have to adapt more, that's when you select for mutator for a lot of mutations, like for high KKS, a lot of things are occurring. When you're more adapted, you see less of these things. Okay, so to conclude, we see some large effect of the strain, which is surprising, but this is the dominant effect. Some effect of the diet, some strain by diet effect, but almost no pure diet effect. We were expecting maybe you could find that, one diet we find all the strains adapt to the same pathway. The only thing that we find popping out in the CHO diet in both strains and not in the Western diet is mutation in a gene of unknown function, unfortunately. So that's a start. Ideally, we would like to go and to do that across multiple backgrounds, but it's a lot of work. So we have to decide what to do, and I'll give you some hints about what we're doing next. And one thing that is interesting is that it looks like the B2 strains we had was more adapted to the mice gut and even more to Western diet. And so these are the strains that are invading the Occidental world where Western diet is propagated, is used massively. So this is going in the good direction, but n equal 1. So we have to do more. Okay, so where are we going from there? So one thing is that here we're killing animals with mice, and I want to limit that at maximum. And so one way to go is that now we're trying to develop some in vitro gut system. It's not perfect, but one step where we have some control, because here we can sample, but we have to wait for the mice to poop to get the samples or to kill the mice. And so here what we need now, what we're doing now and developing with Benoît Chassin in Cochin Institute, is that we're developing a small fermenter in an anaerobic chamber, and we can put a whole microbiota and at least maintain it. It's much shorter term, but here we can see the impact of the food uptake we put and of the microbiota over something like 2-3 weeks. So here it would be more to put different strains and to see their response to that. So we're doing that. Then, as I mentioned, we're trying to, with François Blanca, we're sampling 200 humans. If we manage to get them, because with COVID, people are kind of afraid to come to the hospital. So it's been way tougher to sample things. And so this is another direction we're trying to look to go more in the wild, but with larger number. And the third direction is now, in the past, we had, like I told you, a few genomes. Now we have hundreds of thousands. And now, because of that, we may be able to have genomes sampled in the wild whose divergence is about the same time as the one we have here, meaning a few years. And so as such, we can use those samples now to study what's going on in the wild. We don't have much phenotypic information, but we can at least try to track some selection. And that's the last thing I'm working on. So finally, I'd like to thank the people, because a lot of people have been working on that. So for the early experiment with Lenski and the thermal adaptation was, I mentioned, Al Bennett, Brandon Gould, Tony Long, Richard Lenski, Jeff Barrick, and Dominique Schneider, Sebastian Vilgros, Lisa Codwell. I haven't presented work on that, but then in the lab, the work I presented was done mostly by Alejandro Cauce for the transposon-seq, for the mice experiment, Mathilde Lesca and Adrien Launay, and for most all 3/4 of the mice experiment were done by Mohamed Galiani, who's an amazing PhD student who's been working super hard on that. And he has one major drawback, is that he's an intensive care unit doctor, and so I haven't seen him for the last year and a half. It's Mohamed here, because he's treating people with COVID on a daily basis, and so we hope to finish this manuscript soon. But we've been really limited because of the situation and the super hard work of Mohamed, who which is painful to see because he's really tired after a year and a half of working almost 7 days a week in the hospital. So thank you for your attention, and I'm happy to take any question.